clearly explained about the economic importance of bacteria. Now, Madam will going to explain the another prokaryotic organism, that is blue-green algae. So, blue-green algae is also called as cyanobacteria or cyanophyceae, members of algae. Madam is going to explain. Okay, please you listen carefully. During this time, if you have any doubt, please ask our respected madam. Madam, please you continue.
Yeah, our next topic is cyanobacteria. Again, the name itself shows it is a bacteria. We call them as blue-green algae, and this cyanobacteria includes 2,000 species and 150 genera and 5 orders with a great variety of shapes and sizes. Means there is very much diversity in this class of cyanobacteria. And ecologically, there are 3 major groups in the aquatic environment. They are mostly present in the aquatic environment. And these are photosynthetic prokaryotes that capture sunlight and use chlorophyll and various accessory pigments. So you have to remember cyanobacteria are performing photosynthesis. Cyanobacteria are performing photosynthesis. Okay. And it is commonly found in lakes, ponds, wetlands, streams, etc. And they play a major role in nitrogen, carbon, and oxygen dynamics of many these aquatic environments. Means these are majorly present in aquatic bodies, and there they are performing the photosynthesis, and they are responsible for maintaining the levels of these elements called nitrogen, carbon, and oxygen. And their diversity was achieved more than 2 billion years ago. Means they are very old organisms. And so they belong to division Cyanophyta, and Cyanobacteria, they are called as we discussed, they are called as blue-green algae. So blue, why the term blue means it is because of the presence of a pigment called the word cyno means blue, and bacteria here, cyanobacteria, why we are calling it as bacteria means cyanobacteria means cyno means blue pigment will be there. And bacteria means it acknowledges that they are more close to bacteria than algae. Okay, here we are calling them by 2 names, cyanobacteria and blue-green algae. Okay, cyno means blue and bacteria means they are more closely related to bacteria. And blue and green are pigments. Blue-green algae means blue and green refer to some pigments. Okay, now. Okay. So cells are small, usually less than 10 micrometers. It is the size of the cyanobacteria. They are lacking nucleus because they are prokaryotes. They do not have nucleus. And chloroplasts or other membrane-bound organelles, often with heterosis or akinete spores. Means as they are prokaryotes, membrane-bound organelles are also not there, and they are in blue-green color due to water-soluble accessory pigments. Just now we have discussed blue-green algae. Why we are calling them as blue-green algae means due to presence of some pigments. Example, phycocyanin. And they growth form as unicells, small clusters, filamentous, or large colonies. How they grow? They can grow individually as individual cells, or as clusters, or as filaments, etc. And they are photoautotrophs. Photo means light. Autotroph means they can synthesize their own food. Using light, they are able to synthesize their own food. Means they are carrying out photosynthesis, and they are aerobic organisms. And molecular evidence shows them to be ancestors of algal chloroplasts. So these are the prokaryotes and these are the eukaryotes. And this is the cyanobacteria in a division. This is the cyanobacteria. And as we already discussed, they are microscopic in size, means very smaller in size, and they are found in different aquatic environments. They even live in extreme environments, and extreme environments means which are where the conditions are not normal. And ecologically, 3 major groups are found in the aquatic environments like mat forming species, bloom forming species, and Cyanobacteria. These are the different types of bacteria or major groups which are found in the aquatic bodies. And first, these are the first organisms to have two photosystems and to produce organic material and give off oxygen as a byproduct. In photosynthesis, oxygen is liberated. Even the plants, when they perform photosynthesis, oxygen is liberated. Okay, so similarly, cyanobacteria or blue-green algae, when they perform photosynthesis, oxygen is liberated as a byproduct. Yes, they take the carbon dioxide and they release oxygen. That is why we need plants, because we need oxygen to breathe. Okay, and these are very important to the evolution of the Earth's oxidizing atmosphere. So as we know, these are the ancient organisms. So they are very much responsible for the evolution of Earth's atmosphere. And the importance of this is they are converting atmospheric nitrogen into usable forms through nitrogen fixation. Even in the last class also, we have discussed about nitrogen fixation. What is nitrogen fixation? It is a conversion of this nitrogen or this nitrogen into ammonia or nitrates. So here you can see there is triple bond between two nitrogen atoms, which is very difficult to break. That is why the plants cannot use this nitrogen. And the organisms like cyanobacteria or other bacteria, they are helping in nitrogen fixation. And these cyanobacteria are having pigments. Just now we have discussed, like chlorophyll A, phycobiliproteins are there, phycoerythrin is there, phycocyanin is there, and allophycocyanin is there. So this term blue-green is due to the presence of these pigments, and they look in that color due to presence of these pigments. And storage, this glycogen is the they store glycogen, and cell walls will have amino acids and sugars. So we have seen they can be singular in nature, or they can form colonies, or they can be present in filaments. Or filaments means a stretch of cells which are arranged in a single row, uniseriate or multiseriate. And the filaments are called as trichomes, which is having a row of cells. Here, these are the row of cells which are covered by a mucilaginous sheath around the cell wall. And what is the function of mucilaginous sheath is it protects the cell from drying and involved in gliding. As we know, these cells or this blue-green algae, they are present in aquatic bodies. They glide using this. They will be sticky in nature, and the stickiness is due to mucilaginous sheath, which helps the cells for gliding movement, and they help the cells that like they should not get dried off. And sheath is often colored. So the mucilaginous sheath which is present, it is colored. It can be red color or blue color or yellow or brown color. This is the color of this mucilaginous sheath is due to presence of different properties. Like red sheath is due to presence of acidic condition, blue due to basic, and yellow or brown is due to high salt concentration. And as we discussed, they are
Excuse me,
performing
madam.
nitrogen.
Madam, previous slide, madam. Madam, please, previous slide.
Previous slide.
Please, yes. So in this, madam has explained the sheath is often different colors we are observed in the aquatic ecosystem. Just because of the pigments, scientist Guideco has clearly explained in Oscillatoria sancta, the color of the thallus, the color is changed in red color. In the color, what we can say that the light, whenever the light is exact, light is comes sunlight in color, it is an acidic in nature. Blue color in basic in nature. Yellow brown is high salt in nature. Even though the Guideco scientist has clearly explained the Oscillatoria sancta, it changed the color. The thallus color is changed from one environment to another environment. The phenomena is called as Guideco phenomena. Guideco phenomenon. Guideco phenomenon was observed in Oscillatoria sancta. The blue-green alga member, that is Oscillatoria sancta. Madam, please continue.
Yeah. Yes, sir. So as I discussed, in ocean, they find different colors. So that colors are formed due to this presence of cyanobacteria. That is what is there. And as we discussed, nitrogen fixation is performed by the cyanobacteria, and the specialized structure required for nitrogen fixation is heterocyst. Okay, this is the heterocyst. This is not normal like other cells. These are different or specialized cells. And these cells are responsible for nitrogen fixation. You can see these are larger than the normal cells. Okay, and function is to perform the nitrogen fixation. You can see the cells here. These are all normal cells, and this is the heterocyst. And success due to ability to tolerate a wide range of conditions. So they can tolerate different environmental conditions like marine, freshwater, hot springs. We can see them in different uh, conditions like marine water, freshwater, hot springs, terrestrial, etc. And coming to the heterocyst, these cells are larger than vegetative cells and they look hollow. They are thick walled and they do not allow atmospheric gas to enter, and photosynthetically inactive means other cells are there which perform the photosynthesis, but heterocysts they do not perform any photosynthesis, no carbon dioxide fixation or oxygen evolution. Because there is no photosynthesis, there is no need of any carbon dioxide fixation or evolving of oxygen. Formation of heterocysts triggered by molybdenum and low nitrogen. If these elements are there, they can help in formation of heterocyst. And nitrogen is a limiting nutrient necessary for the production of amino acids. As we study from school, that proteins are building blocks of the cells. So for proteins are made of amino acids. So for amino acids, main component is required is nitrogen. So nitrogen fixation is more important for any cell. Nitrogen, and only cyanobacteria and prokaryotic bacteria can fix nitrogen. So which organisms are fixing nitrogen? You can get a bit like this. Okay, you can get options like the organisms fixing helping in fixation of nitrogen fixation. Okay, cyanobacteria is the one which is helping. Apart from cyanobacteria, yesterday's class we have seen nitrosomonas, nitrobacter also helping in nitrogen fixation. Okay, you have to identify that options. And on of these only two of these two, only cyanobacteria evolve oxygen during photosynthesis. Means the bacteria, that is nitrosomonas and nitrobacteria, also fixing in the nitrogen, but photosynthesis is performed by only cyanobacteria. Okay, so oxygen is evolved by cyanobacteria, not by other bacteria. Important because nitrogenase enzyme is inactivated by oxygen. So why you have to understand this difference of oxygen evolution by these cells is the nitrogenase enzyme which is required for nitrogen fixation is sensitive to oxygen. It is the enzyme. What is the basically what is the enzyme required for nitrogen fixation? You have to remember that is nitrogenase. Okay, and the second point is this nitrogenase is sensitive to oxygen. It is sensitive to oxygen.
Excuse me, ma'am.
Yes, ma.
Excuse me, ma'am.
Yeah, Sabiha.
Ma'am, this
Yes.
bacteria fix nitrogen in soil or in atmosphere also?
Which bacteria, ma?
This
Normal
Nisobacter,
bacteria, nitro.
Cyanobacteria.
Yeah, wherever they are present,
So
they can
they
fix.
stores atmosphere in soil as well as in atmosphere also.
Not in atmosphere. Either they are present in water or soil. What do you mean by atmosphere?
Okay, ma'am.
They
Okay,
cannot
ma'am.
present in air, right? Okay. Right. Important because so these two things you have to remember. Enzyme required for nitrogen fixation and it is sensitive to oxygen. Okay. So sensitive mechanism. Now why we have to discuss that nitrogen is sensitive to oxygen means uh, there is a mechanism to protect this nitrogenase enzyme from oxygen. Okay, means you have to understand nitrogen is having threat from oxygen. So the organism should protect this enzyme from oxygen. So for protecting this enzyme is present in specialized cells called heterocysts. Understood? Where oxygen cannot penetrate into the heterocyst. That is why thick cell wall is present, which do not allow oxygen to enter, and thus this nitrogenase enzyme is protected. And it fixes nitrogen in the dark, but not light, found in non-heterocystic cyanobacteria. So in some other varieties of cyanobacteria, what they will do is, normal cells, they will perform photosynthesis during the day. Okay, but during the night, what happens, as there is no light, there won't be any photosynthesis. So in the night, the same cells will perform nitrogen fixation. Understood? So there won't be any problem with the oxygen. So that time nitrogenase will be active. Here, under aerobic conditions, this is the general reaction for photosynthesis, where carbon dioxide and water, they react, and they carbon dioxide is fixed into carbohydrates. Okay, and oxygen is evolved in the photosynthesis. Electrons for photosystem 1 comes from photosystem 2, which evolves oxygen. When you study about photosynthesis, you will study about these two, photosystem 1 and photosystem 2, how they are, how the electron carriers are there, how they are helping in oxygen evolution at the end of the process. And in the anaerobic conditions, um, carbon dioxide is fixed by, with the help of H2S, and here carbohydrate is formed and sulfur is released. But see, oxygen is not released here. Water is released instead. In the previous slide, oxygen is released, but here you can see water is released. So H2S is the electron donor, so the reaction does not produce oxygen. If there is oxygen here, then oxygen will be released here. But as sulfur is there, here oxygen is not released. And advantages for cyanobacteria is, it can live in fluctuating environments of aerobic and anaerobic with light presence. So when the environment fluctuates between aerobic and anaerobic, I think what is the difference between aerobic and anaerobic conditions. Aerobic means oxygen is present, anaerobic means oxygen is absent. But when the condition changes between these two processes, even then the cyanobacteria can survive. And cyanotoxins in cyanobacteria. So like we have discussed about just now about what was that, food intoxication. Means there we have discussed that bacteria are producing toxins. Here cyanobacteria are also producing toxins called as neurotoxins and hepatotoxins. Again, neuro means these toxins are acting on nervous system, okay, neurons. And hepato means these toxins are acting on liver cells. Hepato means liver cells. Okay, so coming to the neurotoxins, they block neuron transmission in muscles. Example, Anabina oscillatoria. So the neurotoxins are produced by these organisms. They are helping in blocking of neurons. And hepatotoxins means these toxins are produced by Anabina oscillatoria and nostrum. They inhibit protein phosphatase, causing liver bleeding, found in drinking water. Means when the cyanobacteria are present in as a contaminant in drinking water, we should be very careful, because if we consume these hepatotoxins, they are directly affecting our liver by inhibiting this phosphatase enzyme. And swimmers itch is caused by limbia. And movement, how they are moving, they do not have flagella, and or different structures are also not there which help in movement. And they excrete mucilage. In the previous slides we have seen, no, mucilage will be there around the cells, that will help in gliding movement or jet propulsion movement. Helix will have fibers and waves of contraction, some kind of jerky movements they can show by this presence of helix in the cells. And spirulina, this is a filamentous form. It is commonly found in lakes with alkaline pH, and it is a major food for flamingo populations, and it is a commercial food source. Okay, last class when we have discussed about single cell protein, I have discussed about spirulina. Okay, this spirulina is given as a single cell protein. Humans consume this. It can cure many health problems. Okay. Next, Anabina with a heterocyst. This you can see the heterocyst, common bloom forming species with nutrient loads. Whenever you find These aquatic bodies you can find green color. Even at home you can see in water tanks or somewhere. And Limbia martensiana, this releases chemicals which can cause dermatitis. Dermatitis, derma means skin. So it causes problem to the skin. Asexual reproduction is done by hormogonia formation, endospore formation or akinete formation, fragmentation, as well as exospore. All these are methods for asexual reproduction used by cyanobacteria. So coming to the first type, hormogonia. Here, short piece of trichome detaches from the parent filament. See, the small piece will detach from the parent and it will grow into a new filament. Example here, hormogonia. These are the small pieces which can detach. And oscillatoria, here also we can see hormogonia. And next is akinete. Akinete is a thick spore. Akinete is a thick spore which detaches from the filament and it helps in asexual reproduction. See, this is the heterocyst, this is the akinete. In the size, when you compare both, akinete is in larger size. Okay, and these are thick-walled resting spores. Function is to resist unfavorable conditions, appear as large cells than heterocysts, and generally lose buoyancy. Means they do not float in the water bodies. Fragmentation, they cut off from the, like they break into different fragments and then they will grow into new filaments. And cyanobacteria photosynthesize using water as electron donor and produce oxygen. As we have seen the equation there, if water is present, water is reacting with carbon dioxide, we get oxygen as the end product. A small number of strains can also use H2S and convert it into elemental sulfur. This reaction also we have seen where oxygen is not released. And in general, cyanobacteria can tolerate low oxygen conditions and concentrations of H2S that are toxic to eukaryotic algae. Okay, so cyanobacteria are more tolerant to higher levels of H2S conditions or low oxygen concentrations. This tolerance may contribute to their ability to survive in anoxic eutrophic lake sediments as well as in certain matte environments. So this is the property which is helping them to survive at the sediments or base of water bodies. So these are unicellular cells, these are clumps, and this is a filament one. So these are different cyanobacteria you can see. Sometimes this aquatic fern means the plants which are small plants which are present in the aquatic bodies, they have this cyanobacteria, anabena and azole. They are in a relation with this symbiotic relation with these plants, and the large cells are heterocysts. Here you can see the cells which are larger in size, they are heterocysts. And traditional rice farming in many countries involve planting azolla to build up nitrogen concentration. If you have ever visited this rice farming, you can see these plants will be immersed in water. Okay. So cyanobacteria are famous for causing nuisance blooms. They mat growth and many algae like green color is found in this water bodies due to presence of cyanobacteria. So this was about cyanobacteria. If you have any doubts, you can ask. We are having more 3 minutes. Yes, anybody?
Yes, any query, my dear friends? Okay, madam.
Okay.
Remaining bacterial diseases, I will handle, madam. No problem.
Yeah.
Okay, madam.
Okay.
Shall we wind up the today's session?
Yes, yes. Thank
Okay,
you.
madam. Thank you.